Thank you, Ram, for the nice introduction. Good morning, everyone. I will talk about considerations for impurity qualification. The presentation will highlight the key principles of ICH Q3A, Q3C, Q3D, and FDA guidance. 4 cases will also be presented to illustrate the potential pathways for determination of impurity acceptance criteria. First of all, let's warm up with common amplification in the slides. RLD refers to reference listed drug. MDD refers to maximum daily dosage. ITQD refers to identification and qualification threshold. PD refers to permitted daily exposure. The unit is usually in microgram per day. QSAR refers to quantitative structure activity relationship. And last one, PharmTox refers to pharmacology and toxicology. First part of the talk is classification of impurity and the applicable guidance. The case is about MDD selection, which impacts calculation of acceptance criteria. Second part is decision tree and the qualification method of regular impurity. I will talk about justification of impurity specification with RLD samples. Third part is about residual solvent, and last part is about elemental impurity. For topics related to ICH M7, please refer to the presentations by my colleague Barbara and Naomi. I borrowed this slide from Dr. Liao's presentation. Between October 2017 and June 2020, 234 first cycle DMFs have major deficiencies. 83 of major deficiencies are related to qualification of impurity. So how can we qualify impurities in drug substance right at the first time? This slide shows classification of impurities and the applicable regulatory guidance or compendium. Regular organic impurities can be qualified with USP monograph, ICH Q3A, and ANDA guidance for impurities. Residual solvents is under ICH Q3C, and elemental impurities is under Q3D. Qualification of mutagenic impurities will be discussed in Barbara's talk. Several types of drug substances, such as peptides, semi-synthetic, and fermentation products, are not covered by ICH Q3A. The related organic impurities in these types of drug substances are qualified under the guiding principle of clinical relevance. What does clinical relevance mean for impurity qualification? For any impurity in drug substance, establishment of the acceptance criteria is guided by the clinical impact and hazard assessment. It's not by manufacturing process capability. For drug substance excluded from ICH Q3A, acceptance criteria for specified impurity up to the ICH Q3A qualification threshold are generally acceptable, provided that there are no toxicological, immunological, or clinical concerns. The acceptance criteria should be holistically informed by data derived from clinical trials, non-clinical studies, context of use, prior knowledge, publicly available information, and analytical capability. To qualify impurity, the first step is to locate MDD. MDD refers to the maximum amount of drug substance administered per day. MDD impacts calculation of qualification threshold of ICH Q3A and permitted the concentration limit of ICH Q3C, Q3D, and M7. If DMF supports multiple ANDA NDA with various MDD due to different dosage form or route of administration or even clinical setting, highest MDD should be selected for the calculation. So, where to locate MDD? MDD could be found in RLD labeling. Here's an example of how to locate MDD. First, go to FDA homepage and enter the link of Orange Book. Second, search Orange Book with name of active ingredient. Then, locate RLD and find MDD in RLD labeling. The drug substance of this table is mesalamine. The table is generated from the search results of Orange Book. If MDD is not readily available in the labeling, we recommend you work closely with your ANDA or NDA applicant or communicate with FDA by submitting a controlled correspondence. This table summarizes the MDDs for drug substance mesalamine. Due to different route of administration and clinical settings, multiple RLDs are listed in Orange Book. MDD are distinct, ranging from 1 gram to 4.8 gram. Let's say there's one particular DMF XYZ supporting multiple RLDs. Highest MDD, 4.8 gram, should be selected for calculation of qualification threshold, which is 0.05%. In case that drug substance solely supports drug product with MDD other than the highest in the Orange Book, acknowledgement by DMF holder is needed that reference by different product in the future may require different impurity limits. Once MDD is located, qualification threshold can be calculated for regular organic impurities. The table on the top is copied directly from ICH Q3A. Qualification threshold is 0.05% when MDD is above 2 grams per day. For drug substance with an MDD of less than 2 grams per day, qualification threshold is listed as 0.15% or 1.0 mg per day, whichever is lower. Qualification threshold varies when MDD is between 700 mg and 1,600 mg. As shown on the table on the bottom left, qualification threshold for MDD of 800 mg, 1,000 mg, 1,500 mg are 0.13%, 0.10%, 0.07% respectively. For impurities known to be unusually potent or to produce toxic or unexpected pharmacological effects, qualification threshold should be in line with the specific safety level. In addition to ICH Q3A, there are other qualification methods listed in ANDA guidance for impurities in drug substance. The guidance has been revised in year 2009. Method 1. The observed level and proposed acceptance criterion for the impurity do not exceed the level observed in RLD. Method 2. The impurity is a significant metabolite of the drug substance. Method 3. The observed level and the proposed acceptance criterion for the impurity are adequately justified by the scientific literature. Method 4. The observed level and the proposed acceptance criterion for the impurity do not exceed the level that has been adequately evaluated in toxicity studies. Direct comparison to RLD is the preferred method. Other methods will need pharm tox assessment. This decision tree elaborates the sequence how to qualify specified impurity in drug substance with ICH Q3A and ANDA guidance. It's applicable for non-compendial impurity in drug substance, which is within the scope of ICH Q3A. The sequence consists of 4 questions. Question number 1. Is the impurity potentially mutagenic? If yes, ICH M7 applies. There are few exceptions. For example, drug substance is only intended for advanced cancer treatment or itself genotoxic at therapeutic concentration. In this scenario, the impurity can be treated as regular organic impurity. Question number 2. What is the proposed limit? If the proposed limit exceeds qualification threshold, justification is needed. Question number 3. Is the impurity observed in an FDA-approved human drug product at a similar level, or is it qualified by other acceptable approaches? Question number 4. Do toxicity studies confirm no clinically relevant adverse effects considering patient population and the duration of use? If answer is no, the proposed limit should be reduced to a safe level under the principle of clinical relevance. Case 2 provides an example of how to qualify impurities in non-compendial drug substance with comparative analytical studies. Drug substance is within the scope of ICH Q3A and has a MDD of 64 mg per day. Identification threshold and qualification threshold are 0.10% and 0.15% respectively. Origin observed level in generic lot, observed level in RLD, and the proposed limit are tabulated for specified impurities, any unspecified impurities, and the total impurities in the drug substance. Let's focus on justification of the proposed limits. Impurity A is a metabolite. It's also present in RLD at a level of 1.5%. The proposed limit of 0.5% is set to ensure drug substance potency. Impurity B is also detected in RLD. The proposed limit is in line with ICH Q3A qualification threshold. Impurity C is not detected in RLD, however, is predicted to be negative for bacterial mutagenicity by QSAR. It can be controlled as a regular organic impurity. ICH Q3A qualification threshold applies. Impurity D is controlled below 0.10%. It's based on observed level in RLD. Both drug substance and RLD exhibit a peak with the same retention time under a validated HPLC method and present comparable mass spectra for the impurity. The proposed limit of any unspecified impurity is set at no more than 0.10%, which is in line with ICH Q3A identification threshold. The proposed limit of total impurities is set at no more than 2.0%, which is below the observed level in RLD and doesn't exceed the summation of acceptance criteria for individual specified impurities. Therefore, the justification of proposed limits is adequate. We accept the proposed limits. This is the checklist I use in my day-to-day work to assess non-compendial impurity in drug substance. Prior to assessment, search Orange Book and locate MDD in RLD labeling. Calculate qualification threshold of ICH Q3A. Confirm whether the impurities is non-mutagenic or drug substance itself is genotoxic or ICH S9 applies. During assessment, several scenarios are considered. Is the impurity qualified with ICH Q3A? Is the impurity qualified with RLD? If so, compare the proposed limit to the observed level in RLD. Examine analytical methods. Confirm the proposed limit is in line with RLD specification. Is the impurity qualified with scientific literature or toxicity studies? If so, submit internal farm tox consult. I wish this checklist can help you navigate the process of qualifying non-compendial impurities in drug substance. Let's switch gear to residual solvents. We follow ICH Q3C to assess residual solvents. ICH Q3C provides concentration limit for Class 1 solvents and permitted daily exposure for Class 2 solvents. Class 3 solvents have low toxic potential. PDE of Class 3 solvents is above 50 mg per day. For solvents without adequate toxicological data, justification for proposed levels is needed. The justification is subject to our internal farm tox assessment. Higher levels of residual solvents may be acceptable in certain cases, such as short-term or topical application. Justification for these levels should be made on a case-by-case basis. ICH Q3C provides permitted daily exposure for Class 2 solvents. There are two options for setting concentration limit for Class 2 solvents in drug substance. Option 1 is assuming MDD of 10 gram. Option 2 uses the known MDD. Because MDD of drug substance is typically much lower than 10 gram, calculated concentration limit under option 2 can be significantly higher. The limit under option 2 is considered acceptable only if the residual solvent has been reduced to the practical minimum. The limits should be realistic in relation to analytical precision, manufacturing capability, and reasonable variation in the manufacturing process. The following case is about qualification of diisopropyl ether, which is listed as a solvent without adequate toxicological data in ICH Q3C. Diisopropyl ether is used at early stage of manufacturing process in DMF XYZ. Observed level is no more than 5 ppm. However, a limit of 1,400 ppm is proposed by DMF holder, citing a report from National Institute for Occupational Safety and Health. MDD of drug substance is 4 gram. Can we accept the proposed limit? In first cycle, the proposed limit was 1,400 ppm. The exposure up to 5,600 micrograms per day has never been accepted in any FDA-approved drug product. We request DMF holder to tighten the proposed limit based on batch data or provide the corresponding toxicological data. In second cycle, the limit was revised down to 500 ppm. We initiate an internal farm tox consultation. The limit was later found unacceptable by farm tox consultation. In third cycle, the limit was further revised down to 50 ppm. The proposed limit is acceptable. We have to go through 3 review cycles and 1 farm tox consultation to accept the proposed limit of 50 ppm. The review timeline has to be dragged on accordingly. In absence of toxicological data in ICH Q3C, setting accept criterion of residual solvents in consideration of manufacturing capability might be a practical approach. We follow ICH guideline to assess elemental impurities in drug substance. ICH Q3D presents a process to assess and control elemental impurities in drug product. The principles apply to drug substance as well. ICH Q3D lists 3 classes of elemental impurities and other elemental impurities. Permitted daily exposures are listed for Class 1, Class 2, and Class 3 elemental impurities. Other elemental impurities have low toxicity. They are adjusted by other guidelines and regional regulations. This is a periodic table of elements. There are 118 elements. The elements listed in ICH Q3D are depicted in color. ICH Q3D mentions 34 elements in total. The classification of elemental impurities in ICH Q3D is based on toxicity and likelihood of occurrence. It's intended to focus the risk assessment on those elements that are the most toxic and have a reasonable probability of inclusion in the drug product. Here are a few notes for control strategy of elemental impurities. Elemental impurities intentionally added in the synthesis, for example, catalysts, reagents, must be controlled or justified. Drug substance which supports multiple applications with different MDDs or routes of administration will be held to the most restrictive requirements. Risk assessment of elemental impurities is highly recommended for drug substance. Class 1 and Class 2A impurities should be included. Class 3 impurities should be considered in parenteral and inhalation routes if route-specific PDE is less than 500 micrograms per day. How to qualify elemental impurities in drug substance? PDEs of Class 1, 2, 3 elements have been established in ICH Q3D. Control of these elemental impurities are straightforward. For other elements listed in ICH Q3D, PDEs are not established. They are low risk unless specific quality considerations apply. For elements not listed in ICH Q3D, the proposed limit should not exceed the level observed in RLD. Qualification with scientific literature or toxicity studies is subject to pharm tox assessment. This case is about control of elemental impurities in mineral drug substance. Mineral sourced drug substance is usually produced from mined materials by simple production steps in aqueous systems. Elemental impurities from natural contamination and the concentration may vary widely depending on the source of the raw material. Risk analysis based on knowledge of the production process might be infeasible and unreliable. Elemental impurities should be treated like related substance and routinely controlled in drug substance specification. There are different methods to qualify regular impurities, residual solvents, and elemental impurities. The qualification method might impact assessment timelines. For example, qualification of regular impurities with USP monograph, ICH Q3A, or RLD can be assessed immediately, while scientific literature or toxicity studies need pharm tox consultation. Currently, turnover time for the consultation is 3 months. Please bear this in mind when you select suitable method to qualify impurities. At the beginning of the talk, I mentioned that 83% of major deficiencies cited between October 2017 and June 2020 are related to qualification of impurity. This slide lists a few recommendations to some common issues. In case of incorrect qualification threshold, locate correct MDD for calculation of qualification threshold. Regarding failure to demonstrate that same impurity is present in RLD, please identify the impurity in RLD with validated HPLC method and confirm with specific characterization tests such as MS. In case that proposed limit of degradant exceeds RLD limit, please analyze RLD samples with validated and stability indicating HPLC method. Degradants will increase if sample is not properly handled. For solvent without PDE in ICH Q3C, set a limit in consideration of manufacturing capability. This might be a practical approach to facilitate assessment and may avoid farm tox counsel. In summary, impurity qualification has profound impact on adequacy of DMF. The proposed limit can be justified by closely following relevant guidelines. Impurities are qualified under the principle of clinical relevance. Maximum daily dosage is one of key factors in establishment of impurity acceptance criteria. Proposal of practical limit and the selection of appropriate qualification method will facilitate the assessment process. I would like to thank my colleagues for the discussion and comments of this presentation, and thank you for your attention. After this presentation, if you have any questions, please type them into the Q&A box in the bottom right corner of your screen so that we can address them during the scheduled Q&A panel after this session. If you have a question on this presentation after this workshop is over, please send them to the email address provided here by March 19th for inclusion in the follow-on webinar on April 9th. For additional information on this topic, please refer to the listed posters and presentations by my colleagues. Thank you.